All right, everybody, welcome back. Um, we are now going to begin chapter 4, and we are going to be talking about the cell. We're moving up our biological organism organization structure, and we have just gone from atoms, molecules, and whatnot, then moved up to macromolecules. And now we're going to be moving up to cell organelles and the overall cell itself. So in order to understand cellular biology a little bit, we got to kind of break everything down and talk about the different parts of the cell. But one of the things that you really want to understand, and I've said this before, is that cells are the functional units of the body. They're the smallest functional unit of our body. Okay, and so we are a collection of the cell. We are a collection of cells. Um, to be, I won't say exact, but to be as close to that being exact as possible, there is about 37 trillion cells in your body. And it's these cells that are going to be undergoing their own metabolic processes and whatnot that are going to allow our body to undergo its functions and processes too. So we'll break it down for you here as we move through the chapter here. But keep in mind that the more that you understand about the anatomy of the cell, which is the structure, and the physiology of the cell, which is the function, then you will definitely understand a lot of the concepts for this course. So let's start off by talking about the three components or features of a cell, and that's the plasma membrane, the nucleus, and then the cytoplasm. All right, and we'll talk about each one individually here in a moment. But the plasma membrane, all right, think of that as the outer covering of the cell, like the chocolate coating to an M&M. Okay, so it is the outer barrier, but more importantly, it is the limiting barrier. All right, and its specific functions and description of that term limiting, and we'll talk about that here shortly. But basically, it just separates the stuff that's on the inside of the cell from the stuff that's on the outside of the cell. Okay, because the internal environment of the cell has a lot of ongoing processes, and it cannot be bothered by what's going on external to the cell. Okay, and if you notice some structures on the plasma membrane, we have these extensions, and there's three types, and we started talking a little bit about those in lab. All right, you have cilia, flagellum, and microvilli, and we'll break those down. Cilia is like a hair-like appendage. The microvilli are really, really small, short appendages that come off of the plasma membrane. And then you have flagellum, which actually causes movement of the cell itself. Okay, the next structure or feature of the cell is the nucleus. It is the largest structure inside the cell, and much like the cell itself, it has its own covering, and we refer to that as the nuclear envelope. I want you to think of the nucleus as the control center of the cell. Okay, and inside of it, it contains our genetic makeup. All right, the blueprints to us, or our DNA. Right, and much like the inside of the cell, there's a fluid that's inside, or a gel. All right, the nucleus and its inside has a nucleoplasm. Okay, and it's like a fluid type gel-like substance. And then there's the space or the area between the plasma membrane and the nucleus, and that's the cytoplasm. Okay, and the cytoplasm is where we find a lot of stuff. All right, and there's three parts to the cytoplasm. You need to know this. The first part is the cytosol, right? And that's like the fluid-like type of material. Okay, and then we have all the little mini organs, or what we refer to as the organelles. And then we have kind of like the miscellaneous stuff that's found floating in there, and we refer to those as inclusions. Okay, so we're going to talk about these components, and we're going to start off by talking about the cytoplasm. All right, and then we'll talk about the plasma membrane and the nucleus here. All right, so let's start off with the cytoplasm. Okay,
So the cytoplasm, again, is going to be everything that's found outside of the nucleus, all right, but inside of the plasma membrane. That's what PM here is referring to. Okay. So another name for the cytosol is the intra being within, intracellular fluid, right? And it doesn't have the consistency of water. It's a little bit thicker than that. Okay. So anytime you hear me use the term viscous, I am referring to something that's not going to be as, I hate to use this term, as watery as water, but it's going to be a little bit thicker, more like a motor oil, maybe a little bit thicker than the motor oil, all right? But it's more viscous, all right? But it does have a very high water content, as does most of your body. It's just your body, you'll find a lot of water throughout your body. And in the cytosol, we'll talk about this, we have some dissolved macromolecules, so that could be dissolved carbohydrates, dissolved proteins, dissolved fats, lipids, all right? And then we'll find various ions, for example, potassium, sodium, chloride, calcium, all these different charged atoms will be found inside the cytosol, okay? Now, in addition to the cytosol, we'll find the organelles of the cell in the cytoplasm, all right? And these organelles have several different types of functions, and they're relatively complex structures. And we're going to go through what these organelles are, but we break them down into two categories. The first one is membrane bound, okay, and the second one is non-membrane bound. So if you're a membrane bound organelle, that means that you're covered by something, okay? Obviously, we'll refer to it as a membrane. And usually you have a membrane enclosing, all right, this organelle, because that organelle is usually undergoing or performing some sort of function in which it can't be disturbed by anything outside of that organelle. Okay, so it has to have a membrane that is covering it. Okay, so the first type, membrane bound, has a membrane covering, right? It helps to separate whatever's inside that organelle from the cytosol. Right, we're going to go through the different types of membrane-bound organelles. The endoplasmic reticulum, also known as the ER, the Golgi apparatus, sometimes referred to as the warehouse of the cell, lysosomes, peroxisomes, and then one of my favorites, the mitochondria, the powerhouse of the cell. Then the other type of organelle is going to be a non-membrane bound organelle. Okay, so obviously it doesn't have a membrane. Okay, these things are pretty much proteins. Okay, and it includes the ribosomes, the cytoskeleton, the centrosome, which we'll talk about because that has to do mainly with cellular division, and then the proteasomes. These guys like to go around and kind of clean garbage up inside the cell, right? As do a couple of the membrane-bound organelles. And then the final component of the cytoplasm are the inclusions. All right, now, important to understand that inclusions are only staying inside the cytoplasm temporarily. They're not going to stay long. All right, so they come and go. Okay, so they're not considered to be organelles. All right, because of their temporary lifespan there. All right, so like I said, they come and go. Here's that glycogen, and as you know, we've mentioned glycogen before, and glycogen comes and goes depending on the energy needs of the cell or of whatever organism that cell is a part of. Okay, so here's a nice picture, all right, of the structure of the cell, and you can see all the stuff that is going on in and around the cell. All right, so here you have your plasma membrane, and then you have the different types of external features of the plasma membrane. You have this big long projection, and that's a flagellum. Okay, and that actually will move the entire cell. It's like a tail. There's only one cell in the human body that has a flagellum on it, and that's the human sperm cell. And then you've got these shorter kind of hair-like projections, and these are referred to as cilia. And cilia like to move around and move things past the cell. So with cilia, the cell stays stationary, and the cilia move, and they'll move things past the cell.
And then finally, you got your microvilli. And these have a special function, and normally we'll see these projections coming up, and they help to increase the absorption of the surface area of the cell. Okay? You can see there's a lot of stuff going on here inside the cell, in the cytoplasm. We'll go through all these different organelles here, but here's our control center of the cell. All right, our nucleus here. And on the inside, all right, we have our stored DNA. All right, that nucleus is surrounded by that nuclear envelope, so it keeps the internal environment of the nucleus separate from the external environment of the cytoplasm surrounding the nucleus. Okay, so we'll talk about that. Let's talk about some functions of the cell. All right, believe it or not, cells have functions too. And one of the important aspects or functions of a cell is to maintain its shape, okay? Because the shape of the cell can depend greatly upon what type of function it's going to have, all right? And also the integrity of the cell, all right? Because if the integrity fails, the cell becomes weak, it's dying, it's damaged, all right, then it can become non-functional, and then eventually it can actually die and degenerate. Okay, so all these structures that we mentioned before, like the plasma membrane, we have to maintain the integrity of the plasma membrane, because if it starts to fail, then essentially the cell could collapse, things could get inside the cell that aren't supposed to be there and damage the cell and kill the cell, and that's bad. Okay, some of the other functions of a cell is to, obviously, much like us in general as a whole organism, we want to obtain nutrients because we get hungry, okay? And so we need to take things in. Our cell has to do the same thing. It needs to obtain nutrients because it has to use those nutrients to help in whatever function it's serving, okay? And with those nutrients, they will help to build or create these chemical building blocks for whatever purpose. Okay, if it's a bone cell, it needs to help with these chemical building blocks to increase, all right, the integrity of bone. Okay, or if it's a cell that needs to make a hormone, then it needs, all right, those nutrients for energy in order to create that hormone. And just like with anything, as we eat food and drink liquids, we're going to create wastes, where our cells do the same thing. And so our cells need to get rid of those wastes, because if it doesn't, all right, if those wastes accumulate inside, then it can actually damage the cell, disrupt any of the functions or activities that that cell is having to do, and it can cause, it can eventually lead to cell death. And of course, we don't want that. All right. And of course, our cells are capable of cell division. Not all of them, okay, but some of them are able to divide. Okay, those that divide, that can't divide, are what we refer to as amitotic, and we'll talk about some of those cells later on, right? But obviously, all right, like our skin cells, we need those to undergo division, especially when we're growing, all right? And we want to make more cells of the same type, right? So we are able to grow from an infant to a toddler to a child to a teenager to an adult, right? But also, if we damage cells, all right, if they get damaged, then we actually need to replace them. So what are the three main structural features of the cell? You need to know all three of those: plasma membrane, nucleus, and cytoplasm. All right, so let's jump into the first type of organelle that we are going to find inside the cytoplasm. And we're going to start off with our membrane-bound organelles. And this is the endoplasmic reticulum. All right? I want you to think of the endoplasmic reticulum as like a tubular network, okay, that we're going to see inside these cells. All right? And for the most part, this tubular network, or the endoplasmic reticulum, will be found in close proximity to the nucleus. Right? Not all of it, because they can move around, all right, but most of it will be close. Okay? And there's two types of endoplasmic reticulum. If you look here in the picture, all right, you've got this grouping of the endoplasmic reticulum, all right? It doesn't have any bumps on it. And this one here has bumps. So the one with the bumps, That's referred to as the rough endoplasmic reticulum. Those bumps are ribosomes, and ribosomes help with the synthesis or the building or making of proteins. Okay, and then the smooth does not have any ribosomes on it. So, all right, we'll talk about the functions of these here in a moment. So let me swing over to this slide here. Okay, and you can see with the rough endoplasmic reticulum, we're going to have ribosomes present, so we're going to build and make proteins. So when you see ribosomes, I want you to think immediately of protein production. All right, ribosomes, their job is to make these proteins. Okay, so we're going to see cells that have to make proteins, all right, for whatever purpose. Okay, you're going to have proteins that are going to be used for digestion. So like in some of the pancreatic acid in our cells, if we're going to be making lipase and amylase, those are enzymes that break down fats, all right, and they also help to break down proteins. Right, those cells are going to have a lot of rough endoplasmic reticulum. Beta cells are the cells in the pancreas that produce insulin. Those also will have plenty of rough endoplasmic reticulum because we need to make all those proteins there. Also, in the rough endoplasmic reticulum, we're going to see peroxisomes. They're a really cool organelle. I'll talk about them shortly here in just a few moments. All right, but just keep in mind that we're going to see those, all right, in the rough endoplasmic reticulum. All right, the other type of endoplasmic reticulum that we're going to talk about is the smooth. So they're absent of ribosomes. Okay, and so what their functions are going to incorporate are going to be a lot of synthesis. They're going to be making some stuff. They're going to be helping with the movement of things throughout the cell, and very importantly, the storage of lipids. All right, and phospholipids are important because they make up your plasma membrane, or a good amount of it. All right, the majority of the lipids that are found in your plasma membrane are going to be made up of phospholipids. Okay, and the smooth endoplasmic reticulum, if we are in the liver, okay, and if we're in skeletal muscle, we will see carbohydrate metabolism. We're going to make glycogen there because those are the two tissues and organs that are going to contain glycogen. Right? When you ingest drugs, and I'm not saying, well, yes, illegal drugs, but also prescribed drugs, right? Those drugs have to be broken down in order to be activated in some instances, and so the smooth endoplasmic reticulum will be involved in that. We'll see the detoxification of drugs there, alcohol. So on a big night of drinking, like New Year's Eve or the night before Thanksgiving, all right, a lot of folks, the hepatocytes are going, their smooth endoplasmic reticulums are going to be very busy, all right, the next day or several hours after they've started drinking, detoxifying all that alcohol that they've consumed, right? So we'll see a lot of smooth endoplasmic reticulum found in the liver, right? Because the liver does actually have a series of diverse functions in many different systems throughout the body. Okay, so this slide here is basically kind of like a repeat. All right, from the previous slide again, the rough endoplasmic reticulum. We're going to see a lot of protein production going on in here. Okay, and consider this, and this is one of the things I haven't really stressed in previous semesters, but I have been finding that I've been stressing it more and more lately. So when a protein is going to be made here in the rough endoplasmic reticulum, all right, if it's made in the rough endoplasmic reticulum, all right, most likely, all right, these proteins are going to be sent outside of the cell. Okay, so what will happen is they'll be made in the rough endoplasmic reticulum, all right, and then they have two places that they can go to. They can either go, all right, to the Golgi apparatus, all right, and we'll talk about that in a moment, or they will go to the plasma membrane, okay, and we'll talk about that shortly. All right. So again, we already talked about the smooth endoplasmic reticulum, but you can see all the different functions here that I mentioned previously on the last slide here, all right, that the smooth endoplasmic reticulum has a role in. All right, really cool organelle here is the Golgi apparatus, also known as the warehouse of the cell. All right, it's basically a bunch of these flattened sac-like structures that are kind of stacked on top of one another. Okay, and you'll have, all right, let me show you a quick picture. I'll come right back to this slide here. Zoom in. So here's basically our Golgi apparatus here. Okay, and you can see here, all right, these flattened sac-like structures here. All right, on the bottom portion or the fatter area here, we refer to that as the cis face. All right, and the cis face is basically the entrance into the Golgi apparatus. And then at the far end, we have the trans face. So this structure, because of this, these properties here, we say that it has polarity. All right, you've got a cis face and a trans face, different parts or different ends to this organelle, so therefore it has a kind of a polarity characteristic to it. Okay, so we'll have, let me just backtrack here to the previous slide. Here. All right. So that's what we're referring to when we're saying that something has polarity, and we talked about it in one of our lab classes about a cell having polarity. It has a top portion, which is what we call the apical surface or the apical end, and then the bottom surface, which is what we call the basilar surface or the basilar end. That provides that that polarity characteristic. Well, with our polarity with the Golgi apparatus, the cis face is going to be closer proximal to the endoplasmic reticulum, whereas the trans face is going to be at the far end or distal away from the endoplasmic reticulum. Okay, so this is very important that you understand this with the Golgi apparatus. This is where we're going to send proteins that the rough endoplasmic reticulum just made. It's going to send these proteins into the Golgi apparatus, which they'll be modified. We'll add some stuff to it, maybe take some stuff off. Then we're going to pack it up. We're going to pack it up into its own little envelope, all right, what we call a vesicle. And this vesicle is going to be like our shipping container, and we can ship out, all right, the proteins up to the plasma membrane, all right, to be excreted from the cell. Okay, so these vesicles specifically are known as secretory vesicles because these vesicles are going to either release their contents outside the cell, say it's a hormone like insulin, perfect example, or these vesicles will fuse with the plasma membrane because maybe we're trying to repair the plasma membrane or add some things like some receptors into our plasma membrane because we want to upregulate a certain function of the cell. And so we'll stick these items into the plasma membrane. So if something's leaving the Golgi apparatus, it's either going to go to one of two places. Its secretory vesicle is either going to become a part of the plasma membrane, or the stuff in that secretory vesicle needs to be excreted from outside the cell. Okay, so keep that in mind. All right, so that's what this picture is showing you here, and I won't really go into too much detail on this one portion here, but you can kind of see the role here. Here's our rough endoplasmic reticulum. So we're making a protein, all right, and then we package that protein up, and now it's time to send it to the Golgi apparatus because we want to make some modifications to it. It's not ready yet. We need it for something outside the cell, for example. So we package it up, we send it to the Golgi apparatus. It starts to undergo all these different chemical reactions. We add some stuff to our protein, take some stuff off that protein, and then what we're going to do is we're going to package that protein up, and it can do one of two things. It can go right here to the plasma membrane, fuse with the plasma membrane, and then it can shoot out its contents out here outside the cell, or maybe we've got to repair the plasma membrane or add some receptors there into the plasma membrane. It can fuse with the plasma membrane, and then we're going to include whatever into the plasma membrane for whatever our purpose is. Then you can also see here finally, right, I didn't mention this, but this is where our lysosomes come from. All right, in some situations you can see we're going to excrete something here, okay, and in some situations, all right, and we'll talk about this in chapter 17, not this semester, but in 211, in which we talk about how we make thyroxine, okay, that thyroid hormone. And in some cases, we have to actually fuse, all right, a lysosome, which is a kind of it's an organelle that helps to digest things and convert things. We'll talk about it here in the next slide, all right? And we can see that lysosome will fuse with one of these secretory vesicles, and it will do something, and then we can excrete it out. What'd you get? Oh, nice. You both got Dumbledore. All right. So, lysosomes, one of my favorite intracellular organelles because it does a lot. All right. The first thing you need to know is that it is a membrane-bound organelle. And inside, all right, it has these powerful digestive enzymes made conveniently by the Golgi apparatus. Okay. And so lysosomes in a lot of situations will wander around the cell, all right, and if there's some leftover whatever inside the cell, they'll go and gobble them up and just get rid of it. Okay, and in that example that I gave you for the thyroid hormone, in order for a thyroid hormone to be excreted from the thyroid gland, it must first fuse. All right, you'll get into more detail later on with a lysosome, and then that lysosome will cleave off. It's called the thyroglobin molecule. Okay, once that happens, then the cell then excretes what's called the T3 and the T4 hormone, which is a form of thyroid hormone, into the bloodstream to go wherever it's needed for. Right? But just kind of keep in mind that lysosomes pretty much just wander around the cell. They clean up the mess of things that haven't been digested yet or aren't being used. So it's just kind of, it's like if you go to Universal Studios, one of those folks that kind of sweeps up, you know, like the cigarette butts or whatever, right? And they get kind of dispose of that and get rid of it. All right, the next membrane-bound organelle is the peroxisome. Peroxisomes are really cool too because they play a huge role in prevention of cellular infection. And I'll talk about that in a second here. All right, so these guys come right off of the rough endoplasmic reticulum here. All right, but the really cool thing is inside of these organelles, we have what's known as oxidative enzymes. And these oxidative enzymes are just powerful enzymes that help to digest and kill things. All right, so perfect example, hydrogen peroxide is a very potent cell detoxification chemical, and hydrogen peroxide has the potential to destroy some pretty powerful bacteria. So these peroxisomes will fuse with some of these bacteria and then just dump their hydrogen peroxide right onto that bacteria and just slaughter it. Plays a huge role in the process of beta oxidation, and beta oxidation is a chemical process in how we can create energy in the body. All right. Also, we'll talk about it more, not this semester, about lipid synthesis, the making of fats inside the cell. We're going to talk about that. And then our final one here, the mitochondria. Okay, mitochondria, all right, aptly named as the powerhouse of the cell because it synthesizes or makes ATP. All right, and it does so through the process of what we call aerobic cellular respiration. And that aerobic cellular respiration, aerobic, think of oxygen. And if it wasn't for oxygen, all right, then your mitochondria cannot effectively make, all right, the ATP. Okay, so it's really important that you understand. All right, if you want to use the nickname powerhouse of the cell to help you to remember how the cell or what the cell makes, all right, or what its role, or not the cell, the organelle, what the organelle's purpose is or function is, feel free. But just keep in mind the mitochondria, all right, its function is to synthesize or make ATP, and it does so through the process of aerobic cellular respiration, which requires the mitochondria to have oxygen available. If there's no oxygen available, it cannot undergo aerobic cellular respiration, and it cannot make all that ATP. So what organelle function in detoxification? What organelle functions in detoxification and beta oxidation? That would be the peroxisome. And then for energy production, that would be the mitochondria. All right, let's jump into the non-membrane bound organelles, and that's going to be our ribosomes. All right, so these guys are not going to have their own membrane covering them. All right, and so you're going to see them in two places. All right, one, they'll be free floating within the cytoplasm or the cytosol, and when that occurs, then we refer to those as free ribosomes. Okay, and then the other kind is going to be the bound ribosomes, which we saw because they were stuck on to the surface of the endoplasmic reticulum. Okay, so ribosomes are made in a structure inside the nucleus called the nucleolus. Okay, so these ribosomes are made into what we call these large subunits. Okay, so the parts of a ribosome are constructed in the nucleolus, and then those parts get shipped out of the nucleus, and they all get put together in the cytoplasm. And then some will attach to the endoplasmic reticulum, and those bound ribosomes are going to make the proteins that are going to be made for export, all right? Or they will be made to become part of the plasma membrane, or they will be made for the enzymes in those lysosomes. Okay? So keep that in mind. So if you're a bound ribosome, you're going to make proteins that are going to either leave the cell, become part of the plasma membrane of that cell, or make enzymes that are going to go into those lysosomes. The free ribosomes, all right, these guys here, which sit out in the cytosol, they make all the other proteins. So those proteins are pretty much going to just be needed for stuff that goes on inside the cell. All right, so they'll be wandering around there. So here's a cool picture of some ribosomes. You can see that here's an endoplasmic reticulum. So you can see some bound ribosomes here, and then you can see some free ribosomes just kind of floating around. So we have these subunits here, okay? So they're made inside the nucleolus, and then they get kicked out of the nucleus, and then they get put together. And then this structure here, along with some other things, will make proteins. All right, looking at our non-membrane bound organelles, again, we have our centrosome, okay? And when we talk about the centrosomes, it's a paired organelle, okay? And they basically look like a roll of toilet tissue, all right? The paper, the center there, okay? And what these guys do is, for a long time, they didn't really know what the function of a centrosome was, all right? And the only time that you would see it was at the time of cell division. So we've known for quite some time now, but its function is involved in cell division, but what it does is it actually creates this structure called the mitotic spindle. And the mitotic spindle, all right, is going to be basically like these claws. Like here's one centrosome over here, here's another centrosome over here, all right? And they just shoot out, all right, these protein tubules here, and they go towards the center of the cell. And think of it like fingers. And basically what they're going to do is they're just going to grab onto the stuff at the center of the cell and then pull the cell apart. Literally kind of just tears it in half. Okay? So these little fingers are what we call microtubules. All right? And there's those microtubules that help to pull the cell in half to make two new cells. All right? So important role of the centrosomes is going to be cell division. All right, so you can kind of see them here. All right, and when the cell is not dividing, it's just minding its own business, for whatever reason, they lay perpendicular to one another. Okay, so you can see here's one. So when it's time for the cell to divide, these microtubules sprout out. Well, they'll migrate to opposite ends of the cell, and then these microtubules shoot out of them, and they help to split the cell via the formation of the mitotic spindle. All right, and then one of the last non-membrane bound organelles is the proteasomes. Okay, and I love the term here listed. All right, they're the quality control of proteins. Okay, so there are situations, okay, in which, all right, proteins, all right, are going to be made in the cell, and the cell doesn't get everything right all the time. So sometimes we have to check everything, okay? And if a protein is made and it happens to be a protein that's not correctly made, or it just wasn't made to satisfaction, then these organelles will come around and digest them, right? But what happens first is, how does the cell know what protein to digest and what protein not to digest, right? No problem. What we do is we put a tag on that protein, okay? And it's kind of like a person that walks through a store and puts these tags on items that are damaged, for example. Okay, so say a protein's damaged. Well, we mark the protein with this another protein called ubiquitin. All right, and if you get this ubiquitin tag on you, then it's almost like you're marked for death, you're marked for disposal. And so these proteasomes will come around and they will start to digest these proteins. And what will happen, sometimes we'll have cells that will be making proteins that don't have, that weren't completely broken down. Like this picture here shows us, all right, here's a protein, all right, and maybe it was left over from something. So it gets marked with ubiquitin here, and then essentially what will happen is that protein gets funneled down through the center of the proteasome, and then that proteasome will start to just split off each of these little balls here is an amino acid. It'll split off individual amino acids or just chunks of that protein into peptide groups. Okay, because remember, peptides are anywhere 3 to 21 amino acids long. Okay, so it's really kind of a cool way to kind of recycle. Your cells find a way to recycle things here. Which non-membrane bound organelle functions primarily within the cell? That would be proteasomes. And it's kind of an easy one to remember. Well, I should say too easy, but the first 5 letters here, all right, happen to be first 5 letters of protein. Okay. This is the point in which I would normally say, are there any questions? But you guys can't hear me. All right, and then we got our cytoskeleton here. Okay, this is going to be a structure, well, structure or structures that we're going to see inside the cell. And again, when you look at the term skeleton, cyto being cell, the cell skeleton, all right, think of what our skeleton does for us. One, it helps to give us support. So the cytoskeleton offers intracellular support. Okay, it also helps with organization of some of those organelles inside. All right, much like our skeleton helps with organization of some of our organs. Okay, we talked about those centrosomes there and what their role is in cell division. Well, the cytoskeleton helps with cell division through the formation of that mitotic spindle, those microtubules there. And then if we have to move things throughout the cell, well, we can't really rely on things just haphazardly just floating around and hoping that maybe they'll find their way to their destination. So the cytoskeleton plays a role in that. Okay, they'll help with movement of certain cellular materials in throughout the cell and even to the plasma membrane there. Okay, so you'll see these all throughout the inside of the cell, all right, and even on the inside portion of the plasma membrane. And there's three types of cytoskeleton structures. There's the microfilaments, the intermediate filaments, and then the microtubules there. Just to kind of roughly, quickly go through these cytoskeleton elements here. The microfilaments, all right, they're the smallest one. Okay, and we'll talk more about these in chapter 10 when we talk about the muscles, all right, but basically these microfilaments are made up of what's called the actin protein. And think of it like a pearl necklace, and we just take two pearl necklaces and twist them around one another, all right? So these guys here play a huge role in muscle contraction, all right, but they also do help with maintain the shape of the cell. And then also when we undergo cell division there with the creation of, like I said before, the mitotic spindle. All right, the next size up are the intermediate filaments, all right, obviously a little bit bigger. So they're going to be a little bit more rigid, tougher than the microfilaments. Again, they'll offer support to the cell. And then when we want to attach one cell to another cell, we want to hold them in place, right? They offer stabilization between cells, right, at what we call cell junctions, where cells connect to one another. Right? And then the last and largest are the microtubules. All right. So these guys here, all right, are going to be the important thing. Well, first of all, they're made up of the protein tubulin here. All right, but these guys are not permanent. It's very important that you understand that, because, all right, we can change the structure of these microtubulars, make them longer, make them shorter, and it's important that they're not permanent, because when we're transporting things within the cell, if they were permanent, the insides of our cells would fill up fast, and we can't have that, right? Also, when we're engaging in our cell division here, all right, too, when we're forming that that mitotic spindle there, all right, once the cell is divided, we have no more use for that mitotic spindle. So if it were permanent, we'd be stuck with it. But since it's not, we just break it down and get rid of it. Okay. All right. So you can see a couple of examples here, all right, of the cytoskeleton there. Let me quickly go into detail here about the structures of the external surface here of our cell. You heard me mention cilia, the flagella, and the microvilli. The first two have to do with movement. All right. So the cilia are these hair-like projections that sit on the exterior surface of the cell, and what they do is they move back and forth. They kind of beat substances past the cell. All right. So the cell is going to stay stationary. All right. But the cilia will be waving back and forth, and they'll move things past the cell, which is very helpful, especially when we're dealing with the respiratory tract, all right, because you'll have mucus production because you have specialized cells, and we'll talk about more of those cells when we get into the tissue section of this course. But you have these cells that produce mucus, and so the cilia help to move that mucus away so it doesn't clog up or just kind of stick there. The other structure is the flagella. Again, movement, but in this situation, it's going to move the entirety of the cell. Okay, so it's a much bigger projection of from the cell surface there. Okay, and it is the only cell that we see the flagella located is in the human sperm. And then finally, the microvilli. Again, this is another modification there to the external surface. And the important concept with the microvilli that I want you folks to really kind of keep in mind is that it increases the surface area. And that's going to be important, especially when we get to digestion. We have to digest certain substances, all right? As you know, you don't really have a lot of room down there if you look at your abdomen there, but you have your intestines that wind back and forth, and a lot of your intestines are lined by this microvilli, and that helps to increase the digestive surface area even more. Okay, so you can kind of see here, all right, these microvilli. Here's your plasma. Whoops, come on. All right, now instead of having just this small confined area for digestion, now you have all of this area here to digest. So if you were to stretch out the entirety of your small intestine, including all the microvilli here, and open all of that up and lay it flat on a surface, that would roughly equal the size of a tennis court, right? That's immense. That's huge. And it's amazing that we can pack all of that inside of our abdomen, but we do it. All right, so what structure of the cell's external surface is found in parts of the respiratory tract and participates in moving mucus toward the throat? That would be the cilia. All right, I think what I'm going to do is I'm going to stop here because now we're going to enter into the plasma membrane, and that's going to be quite a bit of information for you. And I'm actually going to take a break because my mouth is starting to dry out. So we will come back to the plasma membrane here in our next class. And if you have any questions, you know what to do. Email me or wait till class and we can talk. I hope this video was helpful, and I will see you in class.